Good afternoon, everyone. Thanks for coming to the tutorial from Araceli. My name is Virneliz Fernandez-Vega. I'm one of the senior scientists at the Herbert Werher UF Scripps Institute for Biomedical Innovation and Technology. Part of my talk will start with a brief introduction of our screening center in Jupiter, Florida. I will show you some of our HGS capabilities, and then I will transition in our 3D models that we have done in outdoor facilities with different collaborations with pharma and also with academia. And then I will transition into the high content imaging collaboration that we did with Araceli. Our HGS Center was opened back in 2005 under Scripps Florida Institute. We have a capacity to do cell and biochemical cell-based assays. We have screened over 270 unique targets, and we have identified about 77 molecular probes under NIH-funded initiative. Our funding is mainly driven by NIH grants, but we also have collaborations with pharma and biotech industries. In April 2022, Scripps Florida became part of the University of Florida Academic Health Center, and it was rebranded as the Herbie Werher UF Scripps Institute for Biomedical Innovation and Technology. I wanted to highlight some of the important aspects of our screening center. We have the largest compound library that we own proprietary in-house, that it has over 660,000 compounds. We also have other libraries that, depending on the collaboration, that we can also conduct, like the Mayfish library that is about 15,000 compounds, the FDA, the NCI. We also have a couple of INDs, such as the triple ATPase, P97 target that is being developed by Cliff Biosciences, that is for cancer. GBR6 target from Takeda also is in the IND stage. And we have a drug out there that it was developed by Receptor, was discovered at Scripps, developed by Receptor, and it was later acquired by BMS, which is Ceposia, is to treat MS. Our miniaturization and timing can be completed in a couple of days against our full deck. We have a state-of-the-art technology that allows us to move it that fast. We also are considered part of we are NCI Cancer Institute. Since we become part of UF Florida, now we are considered NCI Cancer Institute, and that also has provided us with other opportunities in which we have this Cancer HTS Drug Discovery Initiative that is allowing collaborators and scientists to focus in cancer targets that can use our HGS capabilities so they can develop their assays and submit a grant that they can come back to us to do the screening. One of the areas that we are focusing a lot in the last couple of years is towards precision medicine. We have conducted phenotypic screening of patient-derived spheroids and also patient-derived tumors on organoids and tested against the NCI library. We wanted to integrate automated platforms that enable rapid and cost-effective solutions looking for to improve personalized treatment therapies. Just to show you our generalized workflow, we can configure any assay that has been transferred to us either in a tube, 96 or 34, and we implement that in 1536, either cell-based assay or biochemical assays. We have a state-of-the-art technology. In the middle, you can see some of those readers, the robotic arm, transfer pin tools, and lately we acquired the Araceli Endeavor that is allowing us to do high content imaging another level very fast. We also, once we complete the screening, we have the chemoinformatics platform that allows us to generate those results back to the collaborators, and also we have capabilities to do LCMS to compound all of the compounds that we tested as part of the screening. Now I wanted to transition and the 3D models. Like I say, we have been focusing a lot in 3D models. We, not only we, but there is enough literature already showing that 3D models provide that bridge between the in vitro and in vivo gap. which means that it will be a better representation, a few representation to do tumor biology and to study the tissue heterogeneity. And that's why it's very important application in cancer research and especially in drug screening. And as you can see in those images, every formation of those 3D spheroids or organoids are different depending on the disease. So the end goal is to accelerate the 3D sphere cultivation using a screening automation so we can improve the efficiency and allow us to rapid do drug testing against libraries that are already out there like NCI and FDA, and we can possibly repurpose or reformulate those drugs out there to treat these diseases or these patients. In vitro cancer disease models are a way and are key to advance to that cancer treatment and personalized medicine. And there are different reasons that probably you're aware: heterogeneity, which I already mentioned, poor prognosis. The lack of appropriate study models is really the key of why we wanted to explore the potential of these 3D organoids to try to improve the drug testing and improve those therapies for personalized medicine. In order to do that, we need 3D technologies. Back in 2018, we developed the first 1536 3D viability assays to assess the cytotoxic effect on drugs using spheroids. This was the first time that we using 3D technologies. In this case, it was a Corning ultra-low attachment plate. We were able to develop this assay. This allows us to use a representative relevant model and support spheroids that will have a better representation to study those models. There's other two technologies that we also have used extensively in the lab, and that's the magnetic 3D bioprinting technology and the 45 angle adapter. Both of them were in collaboration with Greiner. Just to give you a little bit more detail, if you're not familiar, the magnetic 3D bioprinting basically uses a nano shuttle, which is a particle that consists of iron oxide, polylysine, and that will penetrate in the cell membrane. You have to incubate for approximately 14 to 16 hours before you harvest the cells to plate them in these cell repellent plates. And then you put a magnetic drive at the bottom, and that will cause the cells to go all in the center. So that's one way to create these organoids. Another way to create these organoids is with the angle adapter. This was designed also by Greiner, and we could do the 3D printing. This was the last version of it, which basically you just have to harvest the cells, plate them in these cell repellent plates, and just put them in this 45 angle at least for 24 hours, and the cells will aggregate into the corner and will form the spheroids or the organoids. We convert this last technology side by side with the ultra-low attachment. And as you can see in the images in the top right side, we took bright field images at 24 hours and also 96 post-seeding. And as you can see clearly, the formation of the spheres is very comparable between two technologies. This specific cell line that we use for this test, lung cancer cell lines, that has a G12C mutation in the KRAS gene. So we also incorporate drug testing for this comparison, in which we use ARS-1620, which is a novel covalent inhibitor for the KRAS gene. And as you can see, the outcome between the two those responses is very compelling and very comparable with the two technologies. There are different approaches to generate the 3D spheres organoids for high-throughput screening, and these are just some of the approaches that we have used in the lab. One of them is the 2D monolayer cells that were adapted in 2D, but originally those cells were from a human tissue sample called pancreatic. We were able to condition that in 2D and then harvest them and then plate them in those cell repellent plates. We use, at that experiment, we use the magnetic 3D bioprinting. Another approach is using cells that we isolate from the primary glioblastoma tumor biopsies. We isolate it, we keep them all the time in 3D for the GBM specifically case, and we plate them, dissociate them, and plate them in also ultra-low attachment plates. And then the easiest is just to have cells that grow naturally in a 2D monolayer and then dissociate them and then see them in low attachment plates. All of them allow us to be scalable and automation friendly. This is just to give you like a quick overview of our assay protocol that we applied across to all these different cases of studies. Initially, we seed these plates using the optimal cell density, depending on the assay, in a 5 microliter volume using a bioreactor, which is not just a liquid handling system. Again, we use the cell repellent plates. We add the compounds and drugs by using the GNF calypsis transfer system, 10 nanoliter pin tool head, which could give us about 5 micromolar final concentration. We will incubate for 72 hours, and then we will be adding 3D synthetic glow. So it's a very simple phenotypic approach that it will generate luminescent signal. It's just looking at the life and death. So basically, it's just looking at the quantification of ATP produced by live cells in each well. All of this can be completed in less than 5 days from the time that we harvest the sample or we receive the tumor sample by the time of completing the results. I wanted to show you some of the different cases, at least at a glance, that we have generated in the lab. This is the establishment on 3D models of primary human pancreatic adenocarcinoma cells, and also the cancer-associated fibroblasts that were generated from pancreatic cancer patients from the laboratory Dr. Chubisi and Cross at Harvard. In the image that you see on the left is the cells and in 2D. Then the middle bright field image is how these cells form the spheres. And you will tell me probably that these are not spheres, Virneliz. They are. It's just the way that they fan these kind of like field 60 organoids. And as you can see in the maximum projection that we did, we really confirm that these are 3D structures. And it's just the way that they form the organoids. We also tested comparing the 2D monolayer versus the 3D against some well-known cancer oncology drugs. And you can clearly see that the drug sensitivity shifted when you look at the 2D versus the 3D. Again, this is enhancing that there is definitely a need to use 3D model versus the 2D because it will be mimicking what in reality happens in the body. And the need of new therapeutic drugs. In here, I want to show you the capability of cells to form spheroids or organoids. This is a panel of all bright field images that we took at different time points of the pancreatic organoids that we generated. And side by side, we tested with the HT29 and PAC1, which are very well known to use them in cancer research because they form nice spheres. This technology that we use here is using the magnetic 3D bioprinting, side by side with the ultra low attachment. And as you can see, the formation of the organoids is very cell specific. This is showing you that the tissue is heterogeneous and every cell line will form differently. As you can see in panel B, these are the two cancer organoids, HM1 and HG1, completely two different, so pancreatic, and they form completely two different type of organoids. And in panel C, a very low resolution image, but this is an equipment that we developed back in the lab that allows to observe the uniformity and the formation of these figures across the entire plate. This is just like a screenshot of that. Something that I wanted to emphasize that in this type of work, we take a lot of images. And this was not only reading the plate and getting all the images. This is us basically taking each of these plates or these different cell lines and taking pictures at 4 hours, 24 hours, 48, and 72. Now with the Endeavor Araceli high content, we can get the answer in one single read, the images plus the reading. And I will show you that in the later slides. This is another collaboration in which we use lung cancer cell lines. We are interested in looking at small molecules that inhibit that Cas pathway. We had a panel of 10 cell lines in which 5 of them were wild-type Cas, and then other 5 have the mutation G12C sensitivity KRAS mutation. And in here, I'm just showing you 2 of the examples. Again, a lot of images. These are bright field images. 10x after 96 hours plus seeing. You can see the different size of formation according to the cell number that it was seeded. And again, that screenshot that you see at the top is basically, again, using our happy system to visualize. I know it's not very clear. It's a low resolution just to visualize the formation of the organoids formation that we had at the moment. But now that would change now that we have the Araceli in-house, and I will show you some cool pictures later. We wanted to demonstrate that we have the capacity to form 3D cell models with other type of cancer. So we start this project in-house, in which we acquired melanoma and renal carcinoma organoids from the NCI PDMR repository. Normally, they are grown in 24-well plates in 3D dumps. So this is not very ACS amenable. So we do try to adapt them in 2D to make them more scalable, and we were able to do that. As you can see in the first pictures on the left side. Then the next pictures are the plates that were seated. Some of them were seated in cyberplan, some others were in ultra-low attachment. As you can see, the formation cleared of the spheres. And again, side by side, we were always doing the nuclear staining to confirm the 3D structures of these organoids for these two types of models. And again, a lot of pictures, a lot of effort putting all these together that it could change with just one reading with the Araceli Endeavor that I will show you in a little bit. We also wanted to see if we can generate breast cancer organoids. And I have to say, I've been working with organoids for already more than 10 years, and this has been the hardest. One of the samples, we were able to expand them in 3D as they normally grow them in the NCI, and then side by side, play them in a 2D monolayer on top of the matrix gel. And we were able to expand on them. And again, we were able to sit them in these ultra-low attachment plates. And we were able to also engage some of the drug sensitivity of some of the well-known pharmacological controls just to see how they perform. Again, all the data that I've been showing you in terms of the 1536 is scaffold-free. To be HGS amenable, there is no other way that to use a scaffold-free. It would be difficult for us to use any 1536 with matrix gel. I wanted to spend on the next couple of slides talking about a little bit about GBM. This is a current effort that we still have in place, and I'm very enthusiastic about this project because it's still ongoing. Basically, we started this project receiving from a local hospital tissue tumor biopsies from patients that have been diagnosed with GBM. I will then dissociate the cells. I will try to expanding them. And if I have enough cells in the day that I'm receiving that sample, I will quickly assess the cell density linearity, trying to have an idea what is my optimal cell density, doing some of the assays statistics C prime, and then I will immediately run the screening. Again, I just wanted to show you some of the images, bright field images at 20x of the formation of this GBM, and also some of the CSTAT confocal images that we acquired just to confirm the 3D conformation. And GBM, one of the characteristics about these organoids is sphere that it has that necrotic core on the center, and you clearly say that in these samples. As of today, we have evaluated successfully over 60 samples. We were able to continue this collaboration with Dr. Donan and Dr. Rahman over in UF in Gainesville, Florida, in which are continuously receiving samples from them, dissociate them, and then run the cell survival proliferation assay. And then we run against the NCI, and we feed back those results back to them. I wanted to show you quickly of those 60, these are only the 2 examples, but just to show you quickly how these organoids form. In the top, you can see the sample 2699 is a melanoma brain metastasis sample. As you can see, it doesn't form the typical sphere, more amorphous, more like a cell application. And then in the bottom, you see the 2665 correspond to a GBM, more compact, rounded sphere. Again, these are images 72 hours post-treatment, and you already can see the heterogeneity of these samples, even though they're developed from the same disease. On the middle is in a scatter plot, in which it's normalized data. I mean, we don't really have a real control. Normally, it's cells the context of the MSO and the media. We're looking for things that kill the cancer cells. And anything that you see in that sample field that is showing up activity is activity against those compounds. And as you can see from the top and the bottom panel, you already can see difference in the response for each of those samples. For every screen, we do a panel of well-known drugs that allow us to engage the drug sensitivity across each sample. As you can see clearly, we see also differences in this, in the response of these very well-known drugs for each sample, even though definitely there are different patients, but even though from the same disease. So this is indicating that the tumor is heterogeneous and can respond differently to the same drugs, again, indicating the necessity of individual treatment. So this is an current ongoing effort in which we receive samples. We create those in vitro ex vivo 3D dissociation primary tumor cells. We do the drug screening in 3D. We complete that CC determination against the NCI library, and we run those control response. So this is continuously happening every time we get a sample and we're able to do the screening. That's completed in less than 5 days. We then generate those results, and then we pass that to our collaborators in Gainesville, and we provide the cells to create those PDFs that they are currently generating. And they have a pretty cool technology called LEAD that allows to improve the drug delivery to the brain. And this is something that is a work in progress that we are pretty excited to be part of it. So as you can see, I have been showing you different examples of 3D cancer models for HGS that we haven't successfully generated information with automation. We have close to 22 publications around 3D models using HGS. We are pretty excited about that, but we wanted to continue leveraging and developing technologies that allow us to do a more deeper study of those 3D models. And this is just to give you an idea where we are heading out. So again, you're seeing the bright field images taken with our microscope. We generate that cell density linearity with a very simple phenotypic approach. And we have very successfully generating a lot of work across different types of cancer using this very simple phenotypic approach. But what we can do to get a more deep and advanced understanding of these 3D models, that we can monitor the 3D growth. As you probably, if you're in the world of imaging, it's a challenge, right? It was a challenge. Doing confocal microscopic, for example, is to get high resolution images, but it's slow for HGS. In general, imaging 3D spheres can be a challenge for a high contrast perspective reader because it's not scalable. HGS using high condensed training has been constrained because that amount of imaging through output that it doesn't have the capability in order to us to do and full depth screening. But now with Araceli Endeavor, that's feasible. Araceli Biosciences have the Endeavor, which is a very powerful high contrast reader, and it's very recognized because it's very fast. You're still getting high resolution images, 0.7 microns per pixel. It has an advanced optics, a 20x objective. It has multiple wavelengths to do fluorescence. And it has bright fields, so you can also do some cell life imaging. And it's compatible with 96, 384, and 1536. You can also do image analysis with their clairvoyance for and segmentation. And it can read a plate at the speed of a luminescent read. It takes 5 minutes to read a luminescence, and this is what it will take you with the new latest version of Endeavor Ultra in 1536 for all childs in less than 5 minutes. These are examples of different assays that were created by Claudia, one of our experts in high content in the lab, in which we have generated assays like cell painting assays using U2S that you can clearly see sharp and very clear dissociation of those subcellular structures inside the cell. We also do a lot of work with neural algorithm, and you can get very sharp images to do eventually some analysis more clearly. And also, because of the bright field, you can do a time course to monitor that algorithm. So the proposed approach, just as a proof of principle, we wanted to explore the capabilities of the high content reader Endeavor to monitor the growth rate using 3D models. Ultimately, the goal is to implement a more advanced phenotypic approach using live 3D cultures that can reflect the biological morphological changes and cell viability in the presence of drugs. Just to give you a quick overview of what we did to test this. As a similar approach, we use cells that were growing to the monolayer. In this case, we use COS31, which is actually a canine osteosarcoma cell line. I need to have a chance to show you, but we also get samples from dogs, and we also be able to do the screening against those samples, and that's pretty cool as well. So for these tests, again, we harvest the cells, we plate them in cell-repellent plates, we use the angle adapter technology to create those spheres, and we incorporate the acquisition of images over time using the transmitted light. And now that was feasible because it only takes like about 10 minutes to read each plate. So you can do multiple reads and collecting all that data. As an endpoint, as a point of comparison, we did the ATP cell viability assay readout just to compare what we normally do. I wanted to emphasize in the image acquisition because this was not possible before. We were not possible to take pictures. Yeah, you can take pictures, but to image a plate in less than 10 minutes, normally it was taking hours, so it was impossible to do that. So now, what we propose here, we acquired bright-field images using the transmitted light across different times, from the time that we see the cells across 4 days and multiple times on the same day. These were our settings to read the plates, to make sure that we capture most of the well. And you have to remember that these spheres will be towards the corners, so that's where we have to increase that field of view. We're still generating high resolution images, and the reading time is around 10 minutes to read. I wanted to show you this first panel of pictures that, again, all of the pictures that you perceive from now on were generated with the Endeavor. So as you can see, all of the pictures homogeneous, no difference. It is so much easier to look at the data and generate quickly this panel to be able to observe the formation of the 3D formation over time. In here, as you can see, at 0 hours, you can see all of them as spread out. As you move to 5 hours, 10 hours, you already can see kind of like forming that cell aggregation, 20, 23, 28, and then by the time of 48 hours, 68 hours, for the specific case of the cell that we use, it formed beautiful rounded shapes, organoids, spheroids, I should say, for this case. So we were able to monitor the 3D formation over time just using bright-field images. Here I'm showing you all of the different cell densities that we test in the context of time. We went from 250 cells all the way to 4,000 cells per well. And we were looking here at the 23 hours, 48 hours, and 68 hours. As you can see at the lowest cell density, you can see already that by eye, that linearity, which looks smaller, actually moves to that higher cell density. They're bigger, and as you move on time, you can see the formation of those spheres and how the surface area of those spheres increases with the cell number increasing as well. So what can we do now with all those images that we acquired? Like I'd say, Araceli also have an imaging software analysis to maximize your data outputs, and that is called the clairvoyance. And the clairvoyance is a machine learning deep neural net-based AI. And for this case, we plug in cell hosts to help with that segmentation and being able to effectively find those shapes and be able to segment in our spheroids. This is the protocol sizes that we use. Again, the detection method is, like I mentioned, the deep neural net, using as a model the cell pulse detection plus segmentation. And as you can see on the right side, this is the segmentation that was successfully being able to find that main sphere on the corner of the well, and it's doing a fantastic job at doing the segmentation. This is the segmentation across different time points, in which you see on the left panel the transmitted light images, and you can see the segmentation as well on the right side. This is specifically for one of the cell densities, so you won't see that big change in the area because it's just looking at the same cell density, but it's doing a good job at segmentating the 3D spheres structures as early as 23 hours. Just to show you more data around, this is looking at the lowest cell density, 250, 500 cells per well, and the 200,000 cells per well. And this is at the 23, 48, and 60 hours. And again, you can see the provision already at the lowest cell density, and you can see how the pixelization area, or the surface area, is increasing as you increase the cell number, and it's doing a good job at covering pretty much the entire sphere at the biggest, even at the highest cell density. So again, it's doing a good job at the 3D segmentation with these high-resolution images that we acquire with Endeavor. Just looking more closely to the data in the well, comparing the lowest cell density versus one of the highest cell well densities. As you can see, in 5 hours, still it's kind of like spread out. So the formation is not quite clear. In the graph that you see at your right side, it's showing that for the lowest cell density, it's been really finding the spheres up to it has the 20 hours. But if you compare with the highest cell density, it's already finding the spheres in just before the 20 hours mark, because they will form more faster because they are bigger and they will be more aggregated versus the other one are smaller. So maybe the segmentation is not recognizing them as a circular shape. If you look at the 23 hours, again, I'll show you that in the previous slide, is doing the segmentation. But I wanted to add this data at the bottom that is looking at the pixel area that you can get out of the matrix using the clairvoyance. And also we convert that to the area, which is basically, you will see the same trend. It's just the scale would change when you multiply that by the 0.27 microns per pixel that the reader can do. And again, for the lowest cell density, the area kind of stay, about the same, and it kind of like goes slightly off for the longest incubation time, which is expected. And then if you compare that with the highest cell density tested, then it will be the opposite. You will start with a bigger number of pixel area, and then it kind of like it goes down, because then the cells will be more compact together. So again, this is aligned in what we observed before with other studies that we have done for 2D global linearity. One of the capacities that you can do with the clairvoyance, and that you can quickly access some of the data visualization by doing different type of plots. You can do histogram, you can do dose response curve, whisker plots. So it allows you to really look at the data quickly, so you have an idea how your assay perform. On the left is you have the concentration cell number on the x-axis, and you have on the y the spheroid pixel area. This is looking at the mean from the lowest cell density to the highest cell density. And again, it's looking at this is fitted as a single dial fit curve, and you can see that it's doing a pretty good job at that cell density linearity, and is very comparable with our results with the ATP cell viability assay. On the right side is the histogram, in which it's just showing how many spheres we are finding on each well. And for most part, it's finding one sphere in each well, and in some other cases, it's finding like 2 spheres, which aligns with the pictures that I was showing you there. This is using, again, all the data that we generated with the different metrics that we incorporate in this analysis. And this is just looking at all of the cell densities that we tested in the context of time, looking at the pixel area. This is over 230 replicates per condition. And with the early time point, you can see clearly that density linearity, which the lowest sum number have a lower pixel area. As you move up, it will have a higher pixel area. As you move over time, they kind of meet together and show that good reproducibility and that linearity and that expansion of the cells as they move on already formed as a 3D spheroid. So it makes sense for them to have that kind of pattern. So it's expected. And you have to keep in mind that the pixel area is doing 0.27 microns per pixel. So you're pretty much, the distance between every pixel is very close. So this is a very accurate and a very tight data overall. This is looking on the right side at eccentricity. And eccentricity basically is a qualitative measurement that basically will quantify how much of the object that is in the world deviates from a proper circle. Proper circle will be considered to be zero, a zero value. And in here, as you can see in this Bionic plot, I call the 3 time codes, 23, 48 hours, and the 68 hours. And then the 23 hours will be the one here in the far right, in which the early time points, that value, it's more close to 0.8, close to 1, versus when you move to the later time points, those values are more close to a little bit lower, meaning that they are more rounded. Again, all of these if aligned with the 2 sets of data that I'm showing you here. This is another way to visualize the data that you can also generate it in the imaging analysis software of the clairvoyance. This is looking at the cell count that initially was dispatched, but it's a spherochyme, I should say. Looking at the pixel area on the middle panel and the eccentricity. At the early time point, at T0 hours, the dark blue basically means that it's not really finding nothing too much at that early time point. It is finding some with the highest cell density. It goes from left to right, goes to higher cell density. And then as you move to the 23 hours, it's finding more. And then by 48 hours, it's doing a pretty good job finding spheroid. At this one is sphere-ish well. When you look at the pixelization, the pixel area is showing a similar pattern in which is finding some already, is measuring already some of them at the higher cell density. And already at 23 hours, you already can see that linearity and that heat map of difference in the pixel area from the lowest cell density to the highest cell density. And then at 48 hours, it looks aligned with what we expect to see with this cell density. For eccentricity, again, it's having a similar pattern, which is finding already measuring some values at the highest cell density. And then as you move on with incubation, it's doing a pretty good job. It's not that easy to observe. You're talking about 0.5, 0.7 values. But again, it's doing a pretty good job for the most part in trying to measure how rounded are these 3D spheroids. With that, I wanted to highlight some of the take-up, of the presentation. I show you plenty of examples of that we are being successfully integrating high-throughput screening in 3D models to enhance the discovery of novel compounds and trying to find new effective cancer therapies faster and more efficiently. Now with the high contact imaging and being able to redeploy that fast, it's definitely a new way to be able to do other type of assays that it was going to allow us to do live cell imaging in 3D and being able to adopt that in a novel live cell imaging with Endeavor. The bright field images are key. Those high resolutions are going to be key for the analysis part, and also is going to be important to keep in mind that the amount acquisition images time is comparable with any other type of reader that it will take 5 to 10 minutes to read. The combination of the high content analysis along with the cell pose is providing a powerful tool to analyze 3D spheroids, as you can see in that image. But what are the next steps? And you're probably wondering, I mean, we wanted to test other type of cells because as I showed you, not all the cells are perfect 3D spheroids. Some of them, as you can see, they're amorphous. They don't have that perfect circular shape. So we wanted to see if we can investigate around that with the Araceli Endeavor. Ultimately, we wanted to develop a 3D tool that allows us to analyze multiple plates over time, different 3D models for HGS, and being able to monitor that growth faster. Just to show you a little bit more about future directions, we have I showed you plenty of data that we have combined automation with 3D modeling, but now we wanted to be able to complement not only having an endpoint assay, we wanted to be able to generate a more advanced deep model that allow us to provide a deeper understanding of these models and how we can improve that towards precision medicine. We wanted to incorporate maybe a multiplex imaging that will help us to explore what is going on during that time of exposition to the drugs perhaps. We wanted to be able to have a more bulky approach that will allow us to monitor these to treat growth over time. And we wanted to be able to continue working at 3D models, and we wanted to be able to perform a full depth 700,000 columns in 5 days with a high content screening, for example, just cell painting. And that will be possible with Endeavor in-house. Currently, right now, this is being installed in our facilities. See the rod on the middle of the high res, and we will have 2 Endeavors installed that will allow us to read faster. It will reduce the redundancy, and we will generate high resolution images that we can then use for our analysis and being able to generate non-traditional readouts that will help us to discover unique targets with light cell imaging or fluorescent imaging. Doesn't have to be gigs because you can fix them, but it reads so fast that probably you can get more also information just using light cell imaging. So we have more room to determine which way we want to go because of the technology that we have in place. With that, I just wanted to end the talk. I wanted to give a big thanks to Araceli Biosciences to sponsor me and the opportunity to present here today, and to the entire group. I wanted to also thank you to my lab back in Jupiter and all of the different collaborators for the different 3D models and all of the institutions that make this work possible. And with that, I wanted to highlight that you need to stop by at the Araceli booth 2312. They have a very cool live imaging that will generate, and it will scan a plate and do the segmentation of the 3D spheroids, and it could generate those quickly heat map visualization and also those response that kind of like a quick overview of what I show you now, what you can see alive in their good. So make sure that you stop by and happy to answer any question. Thank you for your attention.